So this is our video about meiosis, and meiosis is the process that contributes to the varieties and also produces haploid cells from diploid cells.
So to begin meiosis, let's take an oocyte from a woman, and that basically has 23 chromatids from the mother and 23 chromatids from the father. And that then, during interphase, all those chromatids duplicate to form a tetrad, which basically has 46 chromosomes of 92 chromatids, and that is a tetrad. So during interphase, the cell grows and the chromosomes condense and duplicate into this. And then during prophase, the chromosomes condense and thicken and match with their homologous pairs in order to then cross over. And essentially during crossing over, all the chromosomes switch genetic material with one another to form this. And then the centrioles in the cell move to the opposite ends. So here's one centriole and here's the other centriole. And that's the end of prophase 1.
So after prophase 1 is metaphase 1, and metaphase 1 is when the chromosomes move towards the middle in their pairs. So like there's like 2 for each pair, and they like line up with the metaphase side.
Okay, how about you guys work on this?
And so now that they're on the same side, there will be other chromosome pairs, but I'm just showing this one right now. And the spindles are attaching to them. So once they're up here in metaphase, they'll eventually go to anaphase, which is just the chromosomes being pulled away, the pairs. So now like these pairs like aren't touching each other anymore and they're going onto their own little side. And then lastly, in telophase, the envelope like reforms, the nuclear envelope reforms around them. And so now there's 2 different ones inside it. And then the last thing is cytokinesis 1, where they finally split, and there's a centriole and one of the pairs of the chromosomes in each new cell.
Okay, so just to recap, we started off with the homologous chromosome splitting into 2 different cells, and from there it leads to prophase 2 when the spindles form, which then leads to metaphase 2 when the chromosomes meet in the middle along the metaphase plate in a single filed line, which then leads to anaphase 2 is when the sister chromatids are pulled apart onto each side, which then leads to telophase 2 when the chromatids are fully apart and completely separated, which then leads to cytokinesis 2 is when the cells split into 4 haploid cells with 1 sister chromatid in each. And because of the crossing over, there's the genetic variance for each of the 4 cells, and the linked genes can separate during meiosis, which is very rare, but it can happen if crossing over takes place right in between the two linked genes.